Good morning. So I was asked by Curtis and Jill just to give a quick overview of the types of research that's going on the ocean microbiome in my lab. And so I'm going to start off right away by telling you about an experiment that we did recently. And the experiment involves two marine microbes. One of them is this heterotrophic marine bacteria. It's Regeria pomeroyi, and it's a member of a group of bacteria referred to as the Rosiobacter clade that we work with quite a bit in my lab. And then the second microbe in this experiment is a photosynthetic organism. It's a marine diatom, Thalassiosauris pseudonana. So what we decided to do in this experiment is just very simply grow each of these organisms by themselves, so a pure culture, and then to put those two organisms together. And we're just going to look at RNA-seq data to see what's different under those two conditions. So I just want to mention Bryn Durham, who's a former now PhD student in my lab, did this work, and we've got collaborators from the University of Washington, Shady Amin and Ginger Armbrust. So when we set up this experiment, when the organisms were growing together, there was a mutual dependency. They couldn't grow without each other. And from the diatom's point of view, it's a B12 oxytroph. So it requires B12 for growth. It can't make it itself. And so here it is in B12 limited growth. And then we can rescue that culture. We can either add B12 to the medium, or we can add the bacterium to the medium. So this Rosiobacter, as a lot of its other relatives, actually has a full biosynthetic pathway for B12. And when it's growing in this culture, it apparently releases sufficient vitamins to meet the quota of the phytoplankton. Okay, so from the other side, the bacterium is dependent because it's a heterotroph and we don't add any organic matter into the medium. And so it has to get all of its carbon that's been fixed by the diatom and then released in some form. And actually the same is true for nitrogen as well. So we added nitrate, which the phytoplankton can use. The diatom can use it, the bacterium can't. And so it has the phytoplankton has to release nitrogen in some form that's usable. Okay, so I'm going to just really tell you the data now from the perspective of the bacterium. So it's got about 4,300 genes in its genome, and very simply we just took the RNA-seq data and we just arranged all of the genes in a big table, and up at the top are the ones that were most highly upregulated when that bacterium was growing together in this mutual arrangement with the phytoplankton. And so what happened is, way at the top of the list is a series of 9 related genes, and I'm showing you those here with the other ones that are bright pink and bold. And the genes encoded a transporter system and a degradation pathway for this compound. It's 2,3-dihydroxypropane-1-sulfonate, or DHPS. And so these genes actually bring, this is a trap transporter that brings it into the cell. It's degraded through sulfolactate, eventually to pyruvate, which is a really good growth substrate for the bacterium. And then the sulfur moiety is actually pushed outside the cell with a dedicated bisulfite exporter. So the genes were way at the top of the list because this was really the single most coherent and strongest transcriptional signal that we saw. And so almost all of these are 2 orders of magnitude upregulated. And then over here on the right is just a qPCR demonstration. We picked one of those genes, which is the DHPS dehydrogenase, one of the first catabolic steps, and just showed that we can get the same upregulation either when we regrow it with the diatom or when we just add DHPS as the single carbon source. So when we found this really intense transcriptional signal sort of screaming at us, one of the things we might have said is, what the heck is DHPS? Because in the oceanography community, certainly not a compound that anybody has really been thinking about. But we didn't say that because we actually knew that this organism had this particular pathway, and what we hadn't known was why it had it. And so we knew that it had it because of some work that was done by Alastair Cook, a bacterial physiologist who worked for a number of years on how bacteria degrade sulfonate compounds. He's retired now. He's at Constance University, and this is a paper from his lab laying out the degradation pathway for DHPS. And so as we started working with marine Rosiobacters, he noticed in the genome sequences that there appeared to be orthologs to some of his sulfonate degradation genes. So for about a decade, we'd send him strains as the genomes came out, and then about a year later, he'd send back a paper showing us how these organisms are able to degrade sulfonate compounds, but we just never knew why they were able to do that. And so down here in this DHPS paper, this is a Rosiobacter, and he, relative of our palm, and he had worked at the pathway in that organism too. Okay, so this is a little bit of an aside, but right about the time that we started thinking about DHPS, there was a paper that was published in Nature, so it was February of last year that it was published. And this was a paper that for the first time showed how bacteria can degrade sulfolipids. So there's lots of sulfolipids that are synthesized in photosynthetic membranes of vascular plants and phytoplankton and photosynthetic bacteria, but it had never really been known how it was degraded. There were some theories out there. So these guys are the first ones to show how the polar head group of a most common sulfolipid, this is sulfoquinavose, is actually degraded. And it turns out it's degraded by a pathway that looks very much like glycolysis. So they're calling it sulfoglycolysis. So the shadow pathway down here is glycolysis, and here's glucose with a phosphorus group substituted on it. And it turns out sulfoquinavose is glucose with a sulfur group substituted on it. And so there's this whole parallel series of genes encoding parallel enzymes that basically degrade sulfolipids, or essentially sulfoquinavose. So what we were most interested in, however, is this. DHPS is actually a product of degradation. So this organism can't use DHPS, so it exports it stoichiometrically out into the environment around the cell. And the environment around this cell, because it's E. coli K12, is the gut. And so kind of interesting as we were thinking about DHPS as sort of this novel compound metabolite that may be important currency in the ocean microbiome, this paper comes out and also has DHPS in it as well, but in this case for the gut microbiome. Okay, so I'm going to back up just a little bit and tell you why we did this experiment to begin with, and it has to do with our interest in the role of the ocean in the global carbon cycle. So on an annual basis, the small single-celled phytoplankton in the ocean fix about half of the carbon that's fixed on the planet. And one of the important fluxes of this newly fixed organic matter is actually released from the cells as small labile compounds right into seawater, and then the bacteria grab those up. So the compounds are released because phytoplankton excrete them, because they get lysed by viruses, they get eaten by protists. And this labile DOC loop, where there's a fast processing of organic carbon by bacteria, some of these compounds have half-lives of minutes to hours in seawater. That's actually about half of the organic matter that was originally fixed, so this very fast loop of carbon. It's been of interest for a long time among microbial ecologists, but what we've come to realize recently is that we don't understand as much as we thought we did about what these compounds actually are. So what are these actual currencies that is responsible for this big flux of carbon annually? And so over here on the left is what we thought they would be and what we've been studying as model compounds to learn more about it, and they make sense. You probably come up with that list yourself. But now we've got omics data, and the omics data let us look at what genes are present in these heterotrophic bacteria that are sharing the surface ocean with the phytoplankton, and what genes they're expressing. And so that list now looks really quite a bit different. We certainly have found evidence for these, but it looks like a really very kind of eclectic group of compounds that the bacteria are expressing genes to be taking up. And so down at the bottom here, I added DHPS. So one possibility is that another one of these important labile compounds through which a lot of carbon fluxes in the surface ocean, DHPS might indeed be one of them. But at this point, we identified DHPS in an artificial system with 2 organisms that we put together because they're easy to grow and they've got genetic systems. So probably not a real good way to figure out what's going on in the ocean. And so the next step is actually to move out and see if we can find this process going on as well. And so that means metagenomics and metatranscriptomics. We use a fairly standard process here. I just wanted to point out over here, because we're working in seawater, we typically filter the cells out onto a filter so we can actually, they're more dispersed, and so we can get them into a smaller sample that we can then analyze, either extract for RNA or for DNA. And the other thing I wanted to mention is that we have been using, for the past few years, internal standards for our meta Omics data in my lab. And so we either make artificial mRNAs or we find a genome for an organism that would never be confused with the actual organisms that are present, and we add it in known amounts right onto that filter sample right before we start the extraction. And then those internal standards are carried all the way along in all of the steps. When we get down here and look at the sequence data, we can find those standards. We know what we added. We know what amount, well, how many we found in the sequence library, and so that ratio then lets us look at any particular gene of interest or transcript of interest and then calculate back to that original volume of seawater that was filtered on to the sample. So we're actually getting actual counts of data rather than relative or percent data. And so just to give you an example of what that looks like, this is from my graduate student, Brandon Satinsky, who's doing work in the Amazon plume. So we're looking at a station sort of just offshore that is under a high influence from the Amazon River. And so typically in microbiome research, and Jill has also mentioned this as well, we use different size filters to separate the community into different fractions. And so one of the fractions we looked at is the smaller fraction, cells that'll pass through a 2-micron filter, and that's primarily bacteria and archaea, and some of the very smallest eukaryotic photosynthetic organisms go through. And then another size is the things that are bigger than that, so those are the 2-micron. Those would be mostly all of the eukaryote cells. It would be larger bacteria, and it would also be organisms that are associated with each other or with particles. And so for each one of these in this Amazon sample, we did duplicate metatranscriptomes and duplicate metagenomes, and then we also did a poly-A selected metatranscriptomes. The eukaryotic transcripts hide underneath all the bacterial ones in our samples, and so this helps us actually get a better look at what eukaryotes are in there. And then just to show you here an example of the type of data we can get, and so these are actual either genes per liter or transcripts per liter, and here it's just in very gross categories of bacteria, archaea, eukaryotes, and viruses, but of course for any particular taxonomic group or any particular functional gene, as Matt mentioned, we can actually get counts per liter of genes or transcripts. Okay, so we're going to apply that method now to the DHPS question. And so we were invited to take part in sort of a grassroots organization that sprung up. Ginger Armbrust at the University of Washington headed it up. And the idea was, among a lot of oceanographers, the chemists and the microbiologists, there's been a recognition that there really needs to be better integration of data, even just to the simple fact that we're doing our analysis on the same samples, and that often doesn't happen. So they got together a big group that went out together on a ship, and all different types of chemists and microbiologists. And Bryn Durham, so my graduate student who did the DHPS work, was invited along, and her job was to do metatranscriptomics on this geomics group. So this is Seattle. This is called line P. It's a commonly sampled transect among oceanographers. And we stopped at 4 stations where we did metatranscriptomics. This is P1, P4, P6, and P8. And so Bryn did the same standard metagenomic pipeline that we use, but the first thing that we went looking for in the data was actually one particular gene. We wanted to look for the gene. This is HPSN. That's that first catabolic step in DHPS degradation. And so we wanted to see if we could find evidence that it was being expressed in the ocean. And so I'm showing you here just the single gene data for HPSN, and this is about 25 million transcripts per liter. That's the coastal station, so the one closest to Seattle. And then as we go offshore, the numbers of transcripts drop down. There was somebody on the ship who actually still looks through a microscope and counts and identifies phytoplankton. And so amazingly enough, we also knew what phytoplankton were present at these samples, at these stations. And these are the counts of just the diatoms now. And so it turned out that we were really lucky. This is a place in the ocean that's often dominated by diatoms, but there was actually a nice bloom going on at the time that the cruise was scheduled. And so high numbers of diatoms here inshore and then dropping off as well, sort of in concert with HPSN transcripts. So found evidence that the organisms that we think should be making DHPS and evidence for the bacteria that we think should be catabolizing it are co-occurring along this transect. So we had another member on the Geomics cruise who's a marine chemist from the Woods Hole Oceanographic Institution, and that's Liz Kujawinski. And her job on this cruise was to do metametabolomics. So on those filters that we collected the cells, she took the cells and then extracted this community of microbes to look at what metabolites were present. And so we asked if she could look for DHPS, and she found it, and she actually found quite a bit of it. I'm just expressing this here as percent of about 30 or so metabolites that Liz looked for. And so basically as much as a quarter of the metabolites were DHPS at some of these stations. So we wouldn't expect it to be quite this high throughout the ocean. This in particular was a site that's dominated by diatoms and at this point had a big diatom bloom, but nonetheless evidence that there's DHPS inside the phytoplankton cells, and that connects with the rest of the data that we got. So I've been talking really up to this point just about really one set of genes that were upregulated in this experiment. But we found a number of other transporters for organic compounds that would suggest that they're telling us this labile DOC loop. So what are the compounds that are being released by the diatom and then pulled up by the bacterium? And so I'm showing you here a number of the upregulated transporters for something organic. And the numbers in purple here are actually average fold upregulation of the transporter components. So here's DHPS with that sort of 2 order of magnitude upregulation. The other one I wanted to point out specifically is N-acetyltaurine. So it turns out it's another sulfonate. It's another compound that we knew there was a transporter and degradation system for in this organism, and it was turned up as well in the presence of this diatom. And here's the structure, and you can see this compound actually has a nitrogen a moiety in there, so possibly one of the ways that the bacterium is getting nitrogen from the diatom. And then for the rest of these, some of them we can make a reasonably good guess. A couple of compounds, some we think we know at least what a functional group is, some we really have pretty much no idea of what these compounds are, but sort of a little window, we think, into what these metabolites are that are being exchanged really quite rapidly in the surface ocean between these two microbial groups. Okay, so just a sort of a what are we trying to do when we use omics approaches in the ocean microbiome? What I've showed you, this idea of going to simple model systems to look at specific questions that you clearly can articulate and possibly figure out in a very simple system is really common now. A lot of these organisms that can be cultured have now genetic systems for them. I just recently was at a meeting about diatoms, and there's great genetic systems being developed for them very quickly now with some of the new methods like the CRISPR-Cas. And so this idea that we can actually ask a specific question and get an answer in a simple system, but this is always part of it, the microbiome, ocean microbiome connection, where we've got to go back out to the environment and see if it's a relevant answer to our question, and then this iteration back and forth, and it's really done quite commonly. These are easier because we've got all kinds of tools to do. Jill gave a great example of why that is a lot harder for us to do. The narrower our question, the easier it is, but looking at things like the entire metabolome of the ocean is really quite a challenge for a lot of reasons. So our particular interest has been looking at links in marine carbon and sulfur cycles, and so that's what our questions have often focused on. And then I didn't talk about this, but I just want to mention real quickly, one of the real areas of interest right now in ocean microbiome is sort of something that for people who work with gut microbiomes might not be very surprising, but traditionally, when we first realized that bacteria were important in the ocean, it was assumed that they played a very passive role. So they're the scavengers and the recyclers, and these larger organisms are releasing organic compounds compounds, and the bacteria just go through and use whatever they can. As the omics data started to come in, and actually I have to say it was suspected by other people before they had any data to actually look at it. If you look in the genomes of these bacteria, there's a lot of genes that look like they're homologs to pathogenicity genes. And there's evidence that quorum sensing systems are being turned up, and there's evidence that there's metabolites being released that are analogous to auxin-type compounds. And so this idea that the connection between these autotrophs and this first step in heterotrophy in the ocean may actually be one in which there's a lot of give and take and fight for control and all kinds of things that I think are going to be mutualisms too, that I think will be really very important in the future. Okay, so I wanted to show you Bryn Durham, who is the graduate student now. Both she and Brandon, whose data I showed you, both have graduated now, and they're off doing postdocs. But they did great work in the lab. For Bryn's project, this is Shady Amin, and that's Ginger Armbrust, who are collaborators at University of Washington. They're the Diatom people, so they had the expertise in that level. This is Liz Kudziewinski at Woods Hole, who did our Metagenomics. I didn't show you any data from Sean Campagna, but he's actually here at UT, and he has done metabolomic analysis on the phytoplankton metabolomes, the diatoms. And then just mention and thank our funding sources, NSF and the Moore Foundation. So, thank you.
So this nutritional requirement of the diatom for B12, it's met by the bacterium. Do you think that also happens in situ, or is this an artifact of the laboratory incubation?
No, and that's partly why we set the experiment up that way. It turns out that B12 oxytrophy is extremely common in marine phytoplankton. And so, but some of them have a requirement for it. Some of them don't have a requirement for B12. Some do have a requirement, and they must be getting it from someplace else. And there's good evidence now, again with good chemical data, that you can actually measure B12 and a number of other vitamins also in seawater.
Yeah, that's exactly my point. So vitamin B12 is, you can measure it in seawater. So is there actually a direct dependency of the diatom from a bacterium, or do they just take it up from seawater? So
well, sure.
Right?
Yeah. So it could be either way, but I don't think there's ever a phytoplankton cell in the ocean that isn't surrounded by bacterial cells. So it would be hard to know if they're actually just picking up random molecules that have been released at some point, or whether there actually is a much more efficient way of getting it. So a good question, but yeah.
Maybe a follow-up question. Is this an active mechanism that the bacterium excretes vitamin or corinoids, or is this released when cells lyse?
The bacteria were quite healthy and growing well. It's possible that, of course, some were lysine. But we do know that the bacterium didn't change any of its biosynthetic pathways for B12. And we didn't see any evidence of something that looked like an exporter that had been upregulated.
So it's cool that you were able to find metametabolomic signature that you were able to see in the RNA-seq data. But do you think that will be true for non-blooming situations? So a more, that was sort of like a best case scenario where you have a bloom. How high do you see the resolution going with connecting metametabolomic data to RNA-seq or abundance?
I may not be the right person to answer this, but my impression from the chemists is that if they know the target that they're looking for, they can really measure it down to extremely low levels. So I think if we can tell them what we want to see, that there's a reasonable chance that they can see it. It's not really that they can't see all of these compounds. There are literally tens of thousands of compounds, probably at the same low concentration. And the more important they are, the lower their concentration, because they get taken up so quickly. So if we can tell them what to look for, I do think, yes, that it'll be easy to see it. Not easy. It'll be possible to see it. Yeah.
Steve Wilhelm, University of Tennessee, as always, thought-provoking talk. And just listening to the conversation, the one thing that strikes me is that we don't seem to be considering temporal disconnects between cause and effect, between genes turning on and metabolites being created and disappearing. So do you think that when you find these relationships, you can only find them for strong constitutive relationships, and that the sort of plasticity or flexibility in the genome is being lost?
That's a great question. And I think in some ways we've gotten around it by setting up a model system where we know what's happening, and then second of all, by lucking out on some huge signal on an ongoing diatom bloom. So both of those are true. From every perspective, once we start getting away from a genome into the products of that genome, there's time constraints, there's half-lives of compounds, there's the availability of a substrate even if the enzyme's there, there's all kinds of things, even in a single sample at one time. I think we're really taking a leap of faith here a lot of times. So, yes, I agree.
Hi, Catherine Lemon. Loved your talk. I'm curious with the with the kind of co-cultivation transcriptomics, when you're looking at things, genes whose transcription either increases or decreases significantly, how often do you run across hypothetical conserved open reading frames or hypothetical reading frames?
Yeah.
Because this is something we see in our data, and of course we go after the low-hanging fruit of what is already described
Absolutely.
functionally.
Yeah, which is exactly what I did. There, just because some bacterial physiologist happened to have a love for C2 and C3 sulfonates, we actually were able to solve the puzzle. But there's a lot of things in there. But again, I'm going to say I think there's, in systems like this, there's ways to get at it. And so one example would be we know what transporters we could knock out now. Right? And so we have the genetic system for the organism, and so what we'd love to do is to knock those out, look at the changes in both in the exometabolome and try to see if we can get some better functional annotations. And indeed, some of those, I showed you their transporters,
right.
And so they were all identified as a transporter, but what they're transporting, we have not very good idea for most of them.
Because this thought occurred to me also when Jill was talking about the metagenome work, which is there is, as we amass more and more data from different communities, there may be, I'm curious if there are correlative ways to look for presence of hypothetical genes or their transcription that if we went across multiple different environments would actually generate hypotheses about functions and allow us to begin to get at this, unknowns within microbial genomes.
Yeah.
And one of the things, and I think many people probably have this similar idea, is that some of these may have important functions in the setting of a community and not in primary metabolism, and we're going to find their functions only by looking at a community setting.
Yes. Yeah.
So
I completely agree with that, and I would say probably my wish list for something that I would love to have computationally is a way to do that more easily. I don't really know much about gut microbiome data, but I can tell you the ocean microbiome data is, it would take a year to get the right samples and understand the constraints of them and pull them all down from all these data repositories where they may even just be in raw form, and then to try to ask a question like that. And so that's, I think, a real shame right now, because the data has been at least stable enough now that we could start to put those together.
Because I was wondering, would it be possible computationally to create some kind of clearinghouse, or, it's all the data that never gets published because it's not the main focus of interpreting a story. Some place to start putting that data where you could then take your small sample size and
go
reference
looking
it
for
against a
something
larger
specific.
and see if something that popped up which might allow you to begin to investigate a function.